Okay, thanks everyone for joining us for the Ancestry and Diversity Forum. Today, we are very fortunate to have Dr. Brenna Henn, who is a professor in the anthropology department at UC Davis and also an appointment in the Genome Center. And she's a very established population geneticist who works with African populations. And I'm really excited to hear her talk today. And then I'll let her introduce her student who's also going to be joining. And today, so she'll be taking questions throughout the talk, but we'll have to sort of jump off and we can continue some of the conversation if it's still going, even if she has to jump off. So Brenna, please take it away, and thank you so much for joining us today.
Thank you, Alice. It's great to be here. Hi, everyone. Yeah, as Alice mentioned, please feel free to interrupt me at any point if you have questions. I don't mind that at all. It won't throw me off my rhythm. And I'm going to talk to you today with this I started with a slightly provocative title, which is, why study genetic diversity in African populations? Okay, so a little bit about myself and my lab. My background is in anthropology and population genetics. And so what that means is that I work both on the DNA sampling side of things, which means that we have field sites in Africa, mostly in South Africa, as well as collaborations in Namibia and Ethiopia. And there, we go out, we set up these field sites, we collect DNA samples in cohorts of individuals along with phenotypes. And then we bring the samples back to the United States. And we do a lot of bioinformatic analysis and sequencing and genotyping. And then we also do some methods development, because actually working in African populations, many of the off-the-shelf tools that other geneticists would use they don't perform particularly well in African populations because of their higher genetic diversity. So we have quite a bit of tweaking that we need to do. And we're interested, broadly speaking, in reconstructing demographic history and also quantitative traits. So what is the underlying architecture of these biomedical traits that people are very interested in African populations? So that's mostly what I'm going to talk to you about today, is thinking a little bit about the quantitative trait basis for a few different phenotypes that I'm going to mention. And I wanted to first introduce my student. Hi, Natalie. Can you say hello?
Hello, my name's Natalie. I'm a PhD student in Brenna's lab.
So Natalie will be giving a short flash talk that's embedded within the presentation. It's one of the nice features of the Zoom opportunities, as I get to have my students sort of come in and talk to you a little bit about what they're doing rather than having me summarize it. So she'll do a much better job than I will. Okay, so I started with this slightly provocative title, Why Study Genetic Diversity in African Populations? And presumably most of you here in ClinGen, since the title of this particular working group, I think, is Ancestry and Diversity, you're already convinced that having diverse genetic cohorts is an important thing. But I wanted to briefly discuss why not everybody's actually convinced of this, and it can be quite an uphill battle working as a population geneticist and specifically focusing on African genetic diversity. So I thought I'd start with this quote. This is a real quote from a reviewer in a paper that we submitted just about 6 months ago. And the paper was a methods development paper. So we were presented a new bioinformatic algorithm for basically inferring endogamous population pedigrees. And we applied it to 2 African populations, one which was a large biomedical cohort and the other one which was a small, sort of more anthropologically interesting population. And one of the reviewers responded and said, yes, they are non-white populations, but that alone does not make the solution important. And this paper was rejected not on any technical merit, but because the journal didn't feel like it would be of interest to enough individuals, basically. So, when I read that statement, I was pretty infuriated, because basically it's saying that if I develop a tool for European descent cohorts, I don't need to prove that it works well in non-Europeans. But if I develop a tool for an African cohort, then it's not important unless it also works in Europeans. And so many or some individuals have this perspective, and it's something that I think is really hindering research. And this isn't just a sort of a one-off thing. I gave you a particularly egregious example, but it's certainly not a one-off thing. And I've had these conversations with many other geneticists or researchers in Africa. And the question really is, can we study the genetic diversity in African populations for their own sake? Or does this research need to benefit American or European populations? And this particularly comes up in grant applications, where the burden of selling your grant to the NIH, to the Wellcome Trust, is about making it appear that it will benefit a global set of populations, rather than just understanding the genetic architecture of type 2 diabetes in Ugandans or something like that. So I really want to bring this to the forefront before I sort of begin my presentation, and ask you all to be aware when you're sitting on panel, when you're making these grant decisions, that there is this this sort of hidden burden for those of us who are working in diverse populations, and it's and I think it's a big problem. So and I'm happy to talk more about that after we do some of the research results. Okay, so what are we interested in? We are interested in whether or not biomedical traits, which could be defined very broadly: asthma, BMI, type 2 diabetes, height, whether or not all of this genetic work, which is being done on European descent populations, which have limited genetic diversity, how much is it portable to other global populations? And I've been interested in this question for a long time, but it's recently become sort of more sexy with the development of polygenic risk scores, which I'm sure you're all familiar with. And there's been a lot of work recently asking whether or not these polygenic risk scores are portable to other populations, and by and large showing that there are a lot of complications with this. And so we're really trying to answer this question: is, do phenotypes have the same genetic architecture across populations? And that could mean something related to polygenicity, so how many genes are actually involved. It could be related to complexity, how many variants within these genes are involved. There's a few different ways to kind of frame this question, but basically, or in the polygenic risk sort of terminology, can you predict, given a set of variants, the same phenotype as accurately in a European descent population as you can in, let's say, a South African population? So this is very much an open question right now. There's a lot of great work that's being done sort of more on the theoretical side or on the bioinformatics side, developing and tweaking these polygenic risk score algorithms. So I want to focus a little bit maybe more on what we've discovered looking at particular phenotypes, with a focus on Southern Africa, because that's where I mostly work is in Southern Africa. And I'll particularly discuss 3 examples. So one is skin pigmentation, adiponectin, and then we'll talk a little bit about infectious disease as well. And our goal here is really to understand the complexity and then also the evolution of these traits in African and as well as non-African populations. I see I have one question. Oh, that's a comment. Okay, thanks. Yes, thanks for mentioning the implicit bias issue. Yeah. Okay, so let's just start a little bit talking about the structure of human genetic diversity across the globe. So many of you are probably familiar with this idea of reduced genetic diversity outside of African populations. And there are fairly significant differences in some ways to a population geneticist, they're significant, between populations that live, for example, in South America versus those that live in Eastern Africa. And this is due to the relatively recent and rapid expansion of our species from a homeland, which is probably somewhere in Southern Africa, although it could be more complex than that. And this population certainly was established by 100,000 years ago, probably even more than 100,000 years ago, and then expanded into other areas of Africa first, certainly into Western and Central Africa. And by about 50, 60,000 years ago, there was an established population in East Africa, which exited. And this exit is what we call a founder effect. So there was a very strong reduction in the number of individuals who left Africa relative to the ancestral group living in Eastern Africa. And that caused a reduction in genetic diversity. And you'll see this reduction in genetic diversity occurred as populations continued to bud off and essentially found additional populations into Southeast Asia, Australia, and et cetera. So, there's been a lot of work done characterizing these migration patterns out of Africa, and it's very interesting. I'm going to focus sort of on our ancestral homeland and the populations that still persist there. And just to make this, I think, a really clean and neat example, this is from some work I did as a postdoc in Carlos's lab, where we sequenced about 6 to 8 individuals from 7 different populations around the world, which were meant to sort of capture the breadth of genetic diversity that I just showed you on that previous map. So we had populations from South, Central, and North Africa, Central Asia, Southeast Asia, North Asia, and the Americas. And this is a really simple metric. It's just, we sequence those genomes and then we counted up the number of heterozygotes in each genome. So what you're seeing is the average for each of these 7 populations. And, if you look at Southern Africans or the San, they have about 2.4 million variants that are heterozygous on average, versus you can see this really steep decline all the way down to the Maya, who only have about 1.5 million. So heterozygous variants. So that's a really, that's like a 50%, almost 50% difference in terms of the number that are discovered in the Maya relative to the San population. And that just sort of goes to show you like the amount of complexity that we need to deal with when we're looking at African populations is really exceptional relative to sort of what's been defined for European and East Asian populations. Okay, so where do I work? More specifically, I keep referring to Southern Africa. So I have several field sites across Southern Africa, and I've just sort of given you a sample here. So we started out really working in the Kalahari Desert with the Komani San population. The Komani San are a Khoisan population, and sometimes they're more colloquially referred to as Bushmen. Bushman in the US is considered a derogatory term. In South Africa as well, it can be considered a derogatory term, but I do mention it because that's sort of what most people have heard of. I will say, however, that the Komani San elders don't view this as a derogatory term, and many people do self-identify as Bushmen. But I'm using sort of the more formal ethnic definition, which is Komani San. We've also worked with the Nama, who are also a Khoisan population. They focus more on sheep and cattle pastoralism in the Richtersveld. The Himba, who are agro-pastoralists from far northern Namibia, and these are Bantu-speaking individuals. And then additionally, the Cedarburg, and I'm going to introduce another term, colored. So colored is an apartheid term, just fairly terrible, but again, it's what most people self-identify as. So I'm using it more formally in this context. And these are admixed individuals who have a lot of Khoisan ancestry, but also ancestry from Europeans and Bantu speakers and Southeast Asians as part of the slave trade, which brought many people from South Asia and Southeast Asia to Cape Town. So we have a really diverse cohort of individuals. And we're scaling up. We started a tuberculosis cohort very more recently in the last few thousand years. And the goal is really to grow this cohort to about 10,000 individuals so we can perform sort of proper GWASs. But most of what I'm going to talk to you about today, we're looking at 500 individuals per population, 700 individuals per population, what would be considered relatively small sample sizes for genome-wide association studies. Any questions about that? I threw a lot of ethnic names at you guys very quickly. Okay, like I said, feel free to interrupt. Oh, go ahead, please.
Paired phenotype, what do you actually mean?
So for each individual that we collect, their DNA sample, we also do a demographic interview. So, kind of standard stuff like you would do in a clinic. How old are you? Where were you born? What languages do you speak? Information about their parents and their grandparents. But then we'll also collect information about height, skin pigmentation, and now that we have a more clinical cohort, we're actually able to access the medical records, and then we're getting things like HIV status, verifying tuberculosis, sputum tests. We measure BMI, hip waist ratios, things like that. So it's paired at the individual level.
So Brenna, I'm curious about how you sort of identify your different participants. You mentioned in some cases they speak this language, they're from this region, they're involved in this type of field work. So if from your perspective and then from the study perspective, how are the populations defined and the membership? And then how do you sort of catalog that, and which variables are you using to keep track of diversity or the population membership?
Yeah, that's a great question, Alice. So it does vary sort of across these populations. Like when we started working with the Himba, it was really a partnership with a particular anthropologist, and they're a highly endogamous group. They all speak Himba. They don't really speak any other languages. There's not a lot of sort of in-group migration between different ethnic categories in that region. So that, in that case, it really is an endogamous and relatively homogeneous population. That was very different from when we first started this field site among the Khomani San, and there we really did identify them based on the language. So this community had the last new speakers. So Hnu is a Khoisan language. It's a Southern Khoisan language. And there were, I think, maybe 12 speakers of the language when we first started working there in 2005. And so, from talking to other anthropologists and linguists, we knew that there was sort of this, rapidly dying language up in the Southern Kalahari. But, it was very interesting because when we spoke to other South Africans, they weren't at all interested in them. They said, oh, those people are just admixed. They're not really, they weren't really hunter-gatherers. There's some interesting language, but, they're not like proper Bushmen. They're not like people who, really live up in Namibia and Botswana. And so, we sort of took on the sampling for its own merit. And in this case, because there were only 12 speakers of the language, we didn't just interview the speakers, we also interviewed other members of the community. And so we basically just, worked with our guide, is Magdalena here, and went from house to house. And anybody who self-identified as San or Khomani or Bushman, they could enroll in the study. So we had a very and we never turned anyone away, you know, like if somebody had identified as something else, we wouldn't have turned them away. And so that's, you know, been pretty much my experience working in South Africa, as we go into a particular region or community because of some kind of historical association with Khoisan, or they still have a sort of an active language culture. And then we expand it out. We say, like, you don't have to speak Nama in order to participate in the study. If you only speak Afrikaans, that's fine. You know, if you just identify as a member of this community, we'd love to have you in the study. Is that?
Yes, awesome. Thank you. That's really great.
Thanks, Al. So, you know, this is going to look very, very different from what you may imagine doing sort of clinical DNA collection or phenotyping. You know, we really did interview people sort of underneath these trees. The participant here is sort of being hidden by this acacia tree while her family members look on. And there's some great advantages to doing very small scale sampling. And one of those reasons is that their families are actually able to participate in that process. They're able to see what's going on. If they have objections, they can voice those objections. That individual who is participating can have a conversation. We often, you know, will introduce the study and say, why don't you think about it? And we'll come back tomorrow. You know, if somebody feels uncomfortable or seems a little bit on the fence. And so it really gives us a lot of flexibility, I think, to enroll participants in a really ethical manner and have, you know, consent be a conversation. But of course, it's really slow. And so when we were doing like our skin pigmentation study, you know, it took, I don't know, it took like 5 years to collect 500 people or something. So it's not always possible to continue doing this really artisanal DNA collection if you need to scale up for quantitative traits. So then, you know, we've moved to these other venues in which we'll have like a community meeting. We'll bring everybody in, I'll introduce the project, and then we'll kind of have, you know, a normal sort of, here's the height station, here's the, you know, body mass index station, here's the demographic interview station, and we just go through everybody. I really don't like doing it this way. It's so stressful. I mean, you do get, you know, 30 people a day, but yeah, for me, I much prefer working at sort of a household basis. Okay, so, and how am I doing on time? 9:24, perfect. So I guess, you know, I didn't want to mention the work we're doing without mentioning kind of the ethical side of things. I do spend a lot of time really working with the South African communities, both in terms of the academic community as well as the participant communities, in order to do this. And it's a lot of behind the scenes work that, you know, never really shows up in an academic paper. So we mostly partner with Stellenbosch University, and I'm lucky to have a couple of really great PIs that we work with there for all of our projects. And we've been sort of investing in our relationships there over the last 10 years. And that includes things like co-mentoring graduate students. So we bring graduate students from South Africa to the US. I go and I train people when I'm there, holding workshops. So, you know, all the research is collaborative. If anybody from their group wants to come up to the field with us, we encourage them to do so. We publish papers together. We've applied for many grants together. And we've also pivoted our work so that it more directly benefits our academic partners as well. I mean, that's when I mentioned like this tuberculosis cohort that we've now initiated. That was really because my one of my academic partners, Marla Moeller, that's her primary research interest. And so, you know, we basically said, well, you know, what can we do to benefit you as well, and came up with that particular study. I'll also just mention again, like we work a lot with the participant communities, and they, you know, so there are multiple stakeholders involved, and I unfortunately don't have that much time to get into what do you do with competing stakeholders. You know, I'll say like I have a really positive experience when I work there. almost everybody that we interview, I would say over 90% of the people that we approach want to be part of the study. They think it's really valuable. They are minority populations in South Africa, so they feel like they don't have much of a voice, and participating in scientific studies is a way for them to reclaim their history. It's a way for them to be present in the national conversations that are occurring. So I, by and large, have a really positive experience with the participant communities. We hire RAs from the local communities, even, you know, try to provide more sort of long-term jobs. And then, you know, when we write a manuscript, before we publish the manuscript, we try to make sure that it gets back to the San Council, who is a sort of a political governing council, and make sure that they at least have some input into, you know, the terms that we're using. Like, do we refer to this population as the Komani San? How do we spell Khoisan? That, you know, they'll have input on things like that. Okay, so now sort of finally we get to the science bit, and I'm going to talk about the sort of the first of our three phenotypes. And I initially became very interested in skin pigmentation, because skin pigmentation was thought, if we went back 10 years ago, to be a relatively simple quantitative trait. And by simple, I mean there were probably only a dozen genes that have been identified, mostly in, again, European or East Asian populations. And those genes were thought to have a large effect on the phenotype. So they were easy to measure. You could, you know, genotype 200 individuals, and it's, you know, almost like a Mendelian trait. You'd have a very easy time measuring those effect sizes. And so given how much phenotypic diversity in pigmentation we observed in South Africa, some of you may remember it, South Africa is called the Rainbow Nation. And one of the reasons it's called the Rainbow Nation is sort of the diversity of pigmentation phenotypes that are observed. So, you know, we want to ask this question: are the genes that influence pigmentation in Southern Africans the same as had been identified in Europeans and Asians? Or did they have a different set of genes that influence variation in the phenotype? And so when I started the project, I thought it would be a relatively rapid thing to do. Like, oh yeah, we know what these 12 genes are. We'll just go in, capture them, sequence them. We'll be done. And here, this is kind of one of the underlying evolutionary models I had in mind. So, you know, let's say in Europeans who are sitting out here, there's maybe 3 or some reduced set of genetic variants that are influencing the phenotype that have been selected. Okay, but sort of given what we know about the evolutionary history of Africans, it could be that there is a much larger diversity of genes in Africa, and that's represented by these different colors here that are influencing the phenotype. Now, on top of that, we know that pigmentation is highly correlated with the level of ultraviolet radiation that a population is prehistorically exposed to. So populations that have been in situ at a particular latitude for long periods of time evolve in response to the amount of ultraviolet radiation that they experience in their environment. And so you'll see there's actually this gradation across Africa where there's very high UV radiation, of course, around the equator, but then it's reduced as you move north and it's reduced as you move south. So that, coupled with the idea that we originated in Southern Africa, one hypothesis was that Southern Africans would have, you know, this huge diversity of genes and variants that influence pigmentation. Some subset of them would have been selected for darker pigmentation as populations move closer to the equator. And then as populations moved out of Africa, maybe a few of those made it out of Africa and then were selected for light pigmentation as these populations expanded into very, very high northern latitudes. And so it's possible that some of the variants that were observed in Europeans and Asians actually would still be segregating in Southern Africans and contributing to the phenotype. So that was kind of the model that I had in mind going in. And what we did is we basically collected skin pigmentation phenotypes from about 500 individuals in South Africa. We captured a bunch of really well-known skin pigmentation genes. So by capturing, I mean, we go in and we sequence the full length of those genes specifically. And then we looked at alleles that were known or thought to be known to influence pigmentation in European populations or in other populations around the world. And so you'll see here, like, this is the list of genes that we captured. These are specific SNPs that have been associated with the pigmentation in those genes. And then here are their ancestrally corrected frequencies in these three populations, the Khoisan from South Africa, West Africans, and Northern Europeans for comparison. And you know, the first thing we noted is there was a lot of variation in the allele frequencies. And so we sorted them into these different categories. And the first category basically were, yes, the alleles were present in all of the populations, but actually the allele frequencies varied quite a bit. And you know, you'd see like similar allele frequencies between West Africans and Europeans, even though obviously West Africans and Europeans have pigmentation alleles at the opposite ends of the spectrum. So this maybe suggested to us that those SNPs actually are not probably at the right functional SNPs. I mean, they had been identified as functional, but these things were probably not actually functional in those genes. Then in yellow, we identified alleles that were the light pigmentation allele is high in the Khoisan, it's high in Europeans, and it's lower in West Africans. And so this is the situation which I had sort of hypothesized in the previous slide, where you have ancestral variation and in Southern Africans, there's lighter skin pigmentation variants at higher frequency. Those variants survive the out of Africa bottleneck and are subsequently selected for in Northern Europeans. But then in West African populations, you expect the frequency to be lower because they're being selected against because you want darker pigmentation around high ultraviolet radiation. But this really wasn't the majority of the SNPs that we observed. So actually the sort of the bulk fell into this kind of this gray box, where actually the European variants were simply not observed in the Khoisan. And so these variants are probably things that are de novo in Eurasian populations. They're mutations that occurred probably, you know, after humans left Africa and then were subsequently selected on, but they weren't part of ancestral variation in African populations. And so, you know, we really were able to kind of divide things up into several categories. I see there's a question. So Sharon says
Sorry, it's more a comment than a question, which is this whole idea of trying to find functional SNPs is unbelievably biased towards everyone doing laboratory research who wants to find one, right? So, I mean, I think this is great data, but it just highlights that.
Yeah, you know, some of them had actually been verified in like model organisms, like SLC24A5 was really kind of the first pigmentation gene identified in humans, and they had zebrafish models. So like everybody's super confident that, you know, that SNP is a functional SNP and this gene is contributing to variation in the phenotype. But you're right that there's a lot of different ways to annotate function, and I think some of them probably have worked well, but other ones, you know, many of these have not been, I would say, sort of annotated in like a wet lab manner. They've been mostly sort of bioinformatically inferred to have function, which, yeah, has a lot of biases associated with it. Okay, So then we then just said, well, we're not discovering that many alleles by looking at Eurasian candidate genes, basically. So we're going to go ahead and do a GWAS, and we're going to basically estimate how much of the variation in skin pigmentation is explained by previously known genes and then things that we can discover. So I want to focus your attention on sort of gene set 1. So gene set 1 is what I just mentioned. We basically did a literature review. We pulled out all of the pigmentation genes that had some prior association with skin pigmentation in humans. And then we estimated the heritability in our population. And by the way, the heritability of pigmentation in our population in total is about 100%. So, it's a highly narrow sense heritability defined phenotype. If you just look at those genes, those 15 genes that I mentioned, they only explain about 12% of the variation in the total phenotype. So, so that's not much. That's leaving, that's leaving 90% of the variation unexplained. So then we did a second literature search and we said, well, let's take everything from, mouse studies, zebrafish, basically other candidate genes that have been identified in a variety of different functional and molecular manners. And that really didn't improve things. So here you can see the blue bar here. Again, I think incrementally we got up to about 15% of the variation explained. We then did our own GWAS in this population, and that really improved things up to about 25%. But this was a highly underpowered GWAS, basically, because we only had 500 individuals. And so really, if we put all of it together, and this is talking maybe over 75 genes in total, we're only able to explain about 30% of the variation in this phenotype. So what that's telling us is that this is a highly complex phenotype, much, much more so than in European populations or East Asians. You can, remember when I started out a few slides ago, we said skin pigmentation was thought to be a simple phenotype. It was a dozen genes, maybe a few others, they were of large effect. And that's not what we're seeing in these Southern African populations. There are hundreds of genes influencing pigmentation probably, and most of the genes are contributing a very small effect to the phenotype. So we're currently expanding some of this work, but yeah, the answer here is that the actual architecture of this phenotype is different in Southern Africans. Okay, let's see, about 20 minutes left. So one of the other examples I wanted to give to you, and this is more of an evolutionary story. So there is the Duffy null gene, which is associated with resistance to Plasmodium vivax. And this is the distribution of Plasmodium falciparum today. So vivax is pretty much absent from most of Africa today, but this it was thought historically to have been there. And Sabeti et al. had noted the frequency distribution of the Duffy null allele in the dark gene really mirrored the distribution of present day Plasmodium falciparum within Africa. And they had suggested that this allele had been selected for prehistorically in African populations and facilitated resistance to an unknown malarial parasite, potentially Plasmodium vivax. Okay, so if you look at FYO, which is the Duffy allele, so this is basically the most frequent variant in African populations. Like I said, it's thought there's a lot of sort of wet lab as well as epidemiological work showing that individuals that carry homozygous Duffy null are protected against Plasmodium vivax. And this is what you would look like if you sampled, the 1,000 genomes populations. Everybody in Africa is pretty much 100% Defi null or FYO. And then in Europeans, you have this interesting, almost balanced polymorphism between two other variants. And in East Asian populations, they frequently carry FYA. So they don't have the Defi null allele, they have actually a separate mutation. And if you just kind of looked at this diagram, you probably would have inferred that the FYA, FYB mutation basically occurred in non-African populations and then is being sort of maintained by selection, whereas Defi null was the ancestral allele, and this is what you see basically in all Africans. So then we added two Southern African populations, the Zulu, who are Bantu speakers, and the Omani San, which is one of the groups I just mentioned to you. And instead, what you see is that, in fact, these FYB and FYA alleles are also segregating in Southern African populations. And the work that we did in this particular study suggests that, in fact, Defi null is a newer mutation. So FYA and FYB are actually ancestral mutations. They have been segregating in at least in Southern Africans and the ancestral population for a long time. Then the Defi null allele occurred probably around 60,000 years ago as populations were moving into Central and Western Africa. And then it was very strongly selected for, probably related to malarial resistance, and such that they lost the FYB, FYA haplotype. So you only see the Defi null haplotype. But you're sort of missing, if you had ignored Southern Africans, you're missing all this really interesting and important variation, basically because you'd only focused on 1,000 genomes. And so I'd like to use this example as just a reminder how important it is if you're interested, even in specific phenotypes, to have a wide sample of African populations. African populations are very different from one another, genetically speaking. And so we can't really just take, the 1,000 genomes West African sample and then use that as a representative of all African populations. I think that, let's see, anything else I missed there? Yeah, so this was just standing variation that was lost. It was lost in other Africans for evolutionary reasons. Okay, so let me give Natalie a chance to talk. So one of the ways that we get at genetic architecture without having very large sample sizes is basically combining these two approaches that I just mentioned. So by doing, some work which focuses either on candidate genes or genome-wide association studies, but sort of small GWASs, and then combining that with evolutionary techniques such as selection. And so we're really sort of positive about this hybrid approach moving forward because it allows us to work in populations with relatively small sample sizes. So what I want to talk to you about now, and I'm going to introduce Natalie, is basically an approach to do that. So we scan, you scan the genome of a particular population, and you identify genes or gene ontologies, gene categories that are associated with positive selection. And we've developed this fancy new method to do that called Swiffer in collaboration with Sohini Ramachandran. So Natalie, I'll let you take it away and just let me know when I need to flip slides.
Sure, thank you, Brenna. So one of the projects that I'm currently working on in our lab involves searching for signatures of selection. Specifically, we're looking at for recent balancing selection in the HLA. So what is the HLA? The HLA, or the human-specific MHC, is a part of the genome that underlies a major part of the immune system, which is the processing and presentation of antigens to T cells, which then induces an immune response. This is shown in the first picture to the left here, where you can see this MHC class 2 molecule presenting a peptide that has been derived from the pathogen. And this complex is then bound to the T cell receptor. The HLA is also an extremely unique part of the genome. It consists of an approximately 4 megabase region on the short arm of chromosome 6 that you can see in this figure on the right here. And it's broken up into 3 classes, class 1, class 3 in the middle, and then class 2. And 2 of these classes, class 1 and 2, code for the making of these MHC molecules. It's also a very highly gene-dense region of the genome. It harbors a very high diversity, including many triallelic sites. And it has, on average, a lower meiotic recombination rate than the rest of the genome. And that's mostly driven by the lower recombination rate in class 1 specifically. Next slide, thank you. So selection on this part of the genome is determined by disease pressures that are experienced by population. Like I mentioned before, we're searching for signatures of recent balancing selection. The HLA is a classic example of balancing selection, but most work has focused on long-term or ancient balancing selection. Additionally, we're hoping to elucidate which mechanisms are driving this balancing selection. So for example, is it heterozygote advantage, or more specifically, divergent allele advantage, or instead frequency dependent selection, et cetera. That's, the driving force here. So to do this, we're working with a couple of populations in South Africa that Brenna has mentioned. The Komani and the Nama are recently admixed groups who've experienced recent novel disease pressures that came along with the introduction of pastoralism, agriculture, and colonialism. So we're really focused on the last 2,000 years here. And some of these disease pressures include TB, smallpox, and HIV. Next slide. So to detect any signatures of recent selection, we're implementing this program called Swiffer that Brenna mentioned, and it's a machine learning method that's trained on demographic simulations. And it uses the joint distribution of multiple summary statistics to calculate the probability of selection at a particular site, and controls for false positive selection signals. So on the right, you'll see an example here. The orange and yellow points each represent a simulated population. And if we compare these points, we can see that though we might classify them as being under balancing selection, one of them has a much higher probability of being under balancing selection. The demographic simulations that Swiffer is being trained on are being done in SLIM, which is a forward simulator that also has a coalescent integration. And the simulations include a demographic model and a gene region of interest. And when these simulations are run in the pipeline, you start with a neutrally evolving population, and then you run one as a control, and then in another you introduce the selection. So you have both a control and a selection scenario. And the simulation then outputs a VCF-like file that can be used for the stats, to calculate stats that's used in the Swiffer portion of the analysis. So far, we've been focused on building and perfecting our simulations. We're currently working to implement an HLA recombination map into the simulation rather than just a single recombination rate across the entire simulated genomic region. And we've been performing some sanity checks along the way in the form of 2D SFSs, gene plots. You'll see a gene plot example here to the right, where we were checking to see if our simulation was producing the expected lower diversity within genes that then increases outside of the gene. And as I mentioned before, Swiffer is trained on these simulations. So our next step will be to apply this to some samples from the Komani, the Nama, and the Cedarburg, which we have HLA sequence capture data from to see if we detect selection in response to some of the aforementioned disease pressures. And lastly, I'd just like to say that this project is in collaboration with Dr. Paul Norman and Dr. Sahini Ramachandran's labs. Thank you. I'll turn it back over to Brenna.
Thanks, Natalie. So Natalie gave you an example of sort of what we're doing prospectively moving forward, and I'll just mention, if you look at this particular plot, which is a selection scan on the Komani-san from a couple of years ago, you'll see that the MHC, or the HLA region, you know, it really, really pops out. That's definitely the strongest signal of selection in the genome, but it's incredibly, like she mentioned, incredibly gene dense, and it's hard to kind of parse out exactly what's going on because of the levels of linkage equilibrium in the region and the amount of gene density. And so that's why she's spun off this whole project, which is specifically about sort of untangling these issues specifically for the HLA. So I will, in my last minute or two, just wrap up here and mention we did all these genes, the other genes, the non-MHC genes that are highlighted, are things that have been associated with basically body mass index or metabolism related phenotypes. And so we were able to basically pool all of these selection hits. And one of the things that we noticed, so we pulled all these selection hits, including things like ADIPO-Q and ESRRG, which is, you know, fairly well understood epidemiologically to influence body mass index and adipose tissue. But when we were sort of zeroing in on what's going on, we're like, great, well, we know that there's selection then for this sort of suite of genes that are associated with this, you know, very broadly defined phenotype, BMI or adipose tissue, but sort of like what more specifically is going on, you know, what can we do? So we narrowed in on each of these genes. And again, we have resequence data from individuals in this population for each of these genes. And we identified a missense mutation in the Komani-san, which is at 25% frequency. So you can see here, it's really very frequent in the population. And it, again, is basically completely absent in the rest of global populations that we are able to sample, except for, I think, 1 or 2 individuals from Eastern Africa. That's the 0.5%. So whatever is going on in the San population, it's sort of a unique selection pressure, or it's a selection pressure that is on a particular allele, which isn't present in other populations. So we're now kind of doing like a backwards GWAS, where now that we've used selection to identify which genes are functionally annotated to, we think, be associated with body mass index in the population, we're performing a GWAS on the actual BMI and hip waist ratios from hopefully about 2,000 people. And basically we'll ask whether or not this missense mutation that we discovered can be validated in the sense that, do you see a change in the phenotype for those individuals? So that just sort of gives you a sense of you know, if you pivot, instead of using the GWAS to discover genes, you go the other way around. You use information about selection to first prioritize genes, and then you can do a backwards GWAS from smaller sample sizes. So just to wrap up, you know, I'll mention we can actually do amazing things. Hopefully, I've convinced you we can do amazing things with small genetic population samples. And here, you know, I'm talking 500 individuals, 1,000, 2,000. I mean, things that are really small relative to like the UK Biobank. And we can use these population samples to identify novel genetic architecture. And I think that's especially important in Africans because, you know, what we think we're seeing is that there is a lot of novel genetic architecture there. It's not simply a matter of just porting over genes that have been discovered in Europeans or polygenic risk scores that are discovered in Europeans and then futz with the LD. They have really much more genetic diversity, and that includes greater polygenicity. So more genes are involved in the architecture of these phenotypes than in Eurasian populations. And we can use evolutionary techniques in order to prioritize basically finding, hunting down, functionally validating or doing GWAS studies for these specific genes. And so that really helps with the burden in terms of how many population samples you need to collect to have power to identify these features. But, you know, we need many more individuals. You know, this is true, right, speaking globally, but, you know, I'm sure Alice could also talk to you about how underrepresented they are in GWAS databases. You know, especially from Africa, is just there are so few individuals for which we have genome-wide data. It's really terrible. We need much better phenotypes. You know, you know, you're not working with a lot electronic medical records in Africa. You know, you're working basically right out of paper charts. There's not a lot of consistency sometimes in how people are being phenotyped at these clinics. There needs to be more functional follow-up, and we've got to do a lot better with community engagement as well. So there's a lot of need moving forward to improve this research. So I think there, I'll just pause, and it looks like there are a few questions. So thank you very much.
Thank you so much, Brenna. This was really fascinating and very exciting to see all the work that you're doing. Are there any questions in the last 3 minutes that we have, Brenna? So I guess, Brenna, I'll ask you.
I guess in terms of HLA, one quick question. So I know that the H3 Africa sites have been doing a big HLA project related to infection. Or have you been able to look at any of that data?
No, we haven't actually, unless you have, Natalie.
Nope. Just our HLA sequence data.
Yeah, so we think HLA is going to be pretty important for the tuberculosis phenotype. So for all the individuals that we're doing for our TB case control GWAS, we're actually specifically capturing the HLA region and resequencing that. So we're working with Paul to sort of more specifically find map susceptibility alleles in that region. And I think, yeah, it's like you're saying, this is a known region which is going to be associated with immune susceptibility, so you've got to sort of target it specifically.
No, I understand that. You might get some useful comparisons with the other H3 Africa sites that are doing HLA sequencing. So
no,
this is Jennifer Troyer. I don't know if we've met, but I work with H3 Africa, and I think it just might make a lot of sense to put you in touch with some of those investigators, because in a lot of cases it looks like you're collecting similar types of data, and they, complementary populations potentially. So I think it would make sense to.
Oh yes, please do. That would be great.
Okay, great.
I think there's a question in the chat. Oh, sorry, that's a direct message. Oh, okay. So Laura asks, where did you end up submitting / publishing this paper on methods development? Can you highlight the importance of these investigations to reach editorial boards who may not be as diverse and have some implicit bias? Yeah, I'd love for suggestions on how to do this. We're currently, basically we had a few comments from the reviewers that we think have scientific merit. So we've revised the manuscript based on that. And then we're gonna resubmit to AJHG and just, highlight for the editor some of the egregious comments that were made that really they should have flagged before they sent it back to us and ask them to reconsider it. I'm pretty comfortable pushing back. Maybe it's the, I don't know. I have a relatively thick skin at this point after like years of dealing with this. And so like, I don't mind pushing back on editors. But yeah, I also saw the other day that PLOS is going to have like a PLOS Global Health publication, and Nature has started a Nature Africa publication as well. And so I think it's really nice to see that some of these large publishing houses are sort of specifically, they're aware enough of this issue that maybe these papers aren't sort of making it into their normal venues, that they need to basically do something else in order to facilitate publication. And so I think you're starting to see a little bit of that. But yeah, if you have other suggestions.
Fantastic. Well, Brenna, I know you do have to go at the top of the hour, but thank you so much for joining us today. And I'll connect you with Jennifer Troyer separately offline. That's the beauty of these forums, is that we bring lots of different people together from different parts of the field. So thank you so much for joining us. And I see a comment in the chat, glad you're pushing back. This is from Mildred Cho. I think editors and reviewers could benefit from education about the value of diversity, and a +1 there. So definitely appreciate the work that you're doing, and pushing the frontier forward in population genetics, and hope to have you back at a future date. For those who don't know, Brenda sent me about 3 different talks she could possibly give, and I said, can you give all of them at some point? So we'll hopefully be seeing her and her trainees on future calls.
Thanks so much, guys. Yeah, I'm sorry I have to run, but I have to go teach a 3-hour seminar. So
thank you, Brenna.
Thanks, Carlos. It's nice to see you.
See you guys.
Bye.